Hi there again. Welcome to the 2024 National 5 Biology paper run through. We're going to start off with paper 1 to begin with, and we're going to scroll down to question 1. So, as always, 25 marks, 25 multiple choice questions, and the answers are A, B, C, or D for every question. So, question 1. Cells in the stomach produce enzymes to help digest food. Identify the structure where these enzymes are produced. So this is a tricky first question, to be honest, in the paper because there's a couple of things to consider here. First of all, you have to link two bits of knowledge. One is the 1.1 cell structure knowledge, which is the function of the ribosome, and you also have to link that to topic 1.4 and proteins, where you learn about the five different types of proteins, one of which is enzymes. So you basically have to replace the word enzymes for proteins in this question. And once you say identify the structure where these proteins are produced, it becomes much easier. And that then relies on your knowledge from 1.1, which is that the ribosome is the site of protein synthesis. So that would be the answer for the first one, is C. Question 2. The diagram shows cells viewed under a microscope at a magnification of times 100. Okay, so this is 100 times larger than it actually is. What is the average length in micrometers of the cells shown? So what we have to do first of all is we need to count the number of cells that are going across the field of view here, across the diameter. So that's going to be 1, 2, 3, 4, 5 cells there. Okay, 1, 2, 3, 4, 5. Then we're going to have to do a wee calculation, and we're going to have to get 1200, and we're going to divide that by the 5. That's the 5 cells going across. So 1200 divided by 5 is going to be 240. Okay. Now that gives us the width of, or the length of, one of the cells that are shown, 240 there. And they're asking us for the average length, so that would be fine. Now it says that it's magnified by a scale of times 100 here, but we can actually ignore that because the scale's already in. Okay, question 3. The diagrams represent the movement of molecules across a cell membrane. Which cell would require the greatest number of mitochondria to allow the movement shown? So the knowledge that we have to link in this question is between ATP production and 1.6 respiration, and obviously the function of the mitochondria, which is the site of aerobic respiration. And we also have to link that to active transport, okay, which is our topic 1.2. So, which cell would require the greatest number of mitochondria? Now, it's the cell which is going to need the most ATP, because ATP is produced in aerobic respiration. And the cell that would need the most ATP is the one that's carrying out the most active transport, because as we know from 1.2, active transport requires energy. So if we look at A, we've got a equal concentration both inside and outside the cell, so it's not that one. If we look at B, we've got a higher concentration outside than we do inside, and it moves inside, so that's simple passive transport. E, similarly, we've got a high concentration inside and a low outside, but again, it's passive transport because we're going from high to low. But if we look at D, we're going from a low concentration outside to a high concentration inside, so that would be active transport, and therefore your answer for this one is going to be D. Number 4, a section of DNA contains a total of 6,400 bases, of which 32% are guanine. The number of adenine bases in this section is, so what I always do for this question is I write down my 4 letters, A, T, T, and G, and I write down the numbers. Okay, so if there's a total of 6,400 and 32% are guanine, first of all, let's work out what 32% of 6,400 is. That's 2,048. Okay, and we can do that by multiplying 6,400 by 0.32, 32% as a decimal is 0.32. So 6,400 times 0.32 will give us 2,048. Now, because guanine is complementary to cytosine, that means we also have 2,048 cytosine as well. Okay, so what we do is we times that by 2 to get the total, which is 4,096 between cytosine and guanine. Okay, 4,096. And then we get the total, which is 6,400. We take away the 4,096 and we get 2,304. Now, that 2,304 has to be split between adenine and thymine, which are complementary to each other. So we divide the 2,304 by 2 and we get 1,152. Okay, and that's for both adenine and for thymine. Okay, so the answer is going to be A for this one, for question 4. Okay, so a little bit of maths involved in that one, but we managed to work our way through that. All right, so the key thing is, remember, complementary base pairing, A goes with T, C goes with G. Work out 32% of guanine. You know that cytosine is going to be complementary to that. Then you get that total, 4,096. When you total those two together and you take away from 6,400 and you're left with the 2,304, and then we half that again because we need 50% adenine and 50% thymine from the remainder there. Okay, number 5. Which diagram shows the concentration of product during a degradation reaction? Okay, so for this question here, we look at the graph and we have to work out if it's a degradation reaction. First of all, we are breaking down a substrate into a product. Okay, so a larger substrate into a smaller product. As time passes, a substrate is going to be broken down into more product, so we would expect the concentration of the product to continue to increase over time. Now, obviously, there are circumstances where, for example, you run out of substrate and therefore the product that's made is obviously going to slow down eventually. But this one is during the reaction, so it suggests that there's enough substrate available, which means it's going to be C for this one, where we see that continuous increase in product made over the course of the reaction. Okay, so C for that one. 6, the following stages occur during the process of genetic engineering. We have to put them into the correct order. So if we look at the 4 options: gene inserted into plasmid, plasmid cut open, plasmid extracted from bacterial cell, and gene extracted from chromosome. So we know that the first thing we have to do is we need to locate the gene on the source chromosome. That's not one of the options. Next thing we do is we extract that gene from the chromosome. So that's going to be number 4 there, isn't it? Which is going to come first. We then obviously have to get our bacterial cell and extract the plasmid, then cut the plasmid open. So that's going to be 3, then 2. And then finally we insert our source gene into the plasmid and it becomes a modified plasmid. So that's going to be 4, 3, 2, and 1. So the answer is going to be D there for number 6. You need to know your stages of genetic engineering to help you with that question. So memorize those if you are not sure yet. Number 7, a group of scientists investigated the effect of temperature on the growth of genetically modified cells. The GM cells were grown in a nutrient solution at 20 degrees. This was repeated at 30 and 40 degrees using fresh nutrient solutions at each temperature. Which would the following would improve the reliability of results? So if you watched any of my videos before, you'll know that I always teach the reliability as RIRI. So RIRI is reliability repeat. So we're looking for some sort of repetition here to increase the reliability within this one. So would we set up the nutrient solutions containing GM cells at 25 and 35 degrees? Well, all we're doing there is increasing the number of temperatures that we're using, not actually repeating the experiment more times at the temperatures we already had. So it's not going to be A. B is going to be set up 10 nutrient solutions using GM cells at each temperature. That sounds perfect to me. That sounds like the right answer. C, use the same concentration of nutrient solution at each temperature. That is a good validity step and measure, but it won't improve the reliability. And use the same types of GM cell at each temperature. Again, good for validity, but for reliability, we're looking at B there, setting up 10 nutrient solutions, okay? So that's what we're looking for number 7. Number 8, a respirometer was set up as shown to investigate respiration in maggots. Lovely. Okay, which of the following would be a suitable control for this investigation? So remember, with controls, we are looking for an apparatus that's set up in exactly the same way as the original experiment, but minus the factor that we're investigating, okay? And what we're investigating here is respiration in maggots. So it's likely that every other aspect of the experiment will have to be the same, except from the maggots themselves, okay? So we have a look at the 4 options there. And in D, we've still got the maggots there, but we've lost the the chemical to absorb the carbon dioxide, so that's no good. For C, we've got glass beads to replace the maggots, which is probably quite good for a control. However, we've changed 2 things because we've also lost the chemical to absorb carbon dioxide, so that's no good. For B, we've got glass beads replacing the maggots, which is good. And we've got the chemical to absorb the carbon dioxide, which is also good. And everything else looks very similar to the first one, apart from the glass beads replacing the maggots. So we've changed just one variable there. That's ideal. And then for A, we can see they've switched the chemical and the maggots around, but that's just that's not actually going to work, that experiment there. That we've not actually made that a control there. And what we've done is switch 2 things around in their places. That's all. So the answer is going to be B for this one. Okay, where we've replaced the factor we're investigating, i.e. the maggots, and them carrying out respiration, and we've replaced them with these glass beads here and kept everything else the same. Okay, number 9, a chemical called colchicine, I don't know if I pronounced that right, stops cells from producing spindle fibers during mitosis, which prevents further stages occurring. Which stage of mitosis would still occur when colchicine is present? Okay, so if it stops the cell from producing spindle fibers, which stage is that going to affect? So the chromosomes move to opposite poles of the cell? I would think so. The chromosomes been pulled apart? Absolutely. Those will be affected if the spindle fibers are not there, I should be saying, sorry. So A and B need the spindle fibers. The chromosomes shortening and thickening. Now, the shortening and thickening of the chromosomes does not require the spindle fibers. So I'm thinking at the moment that's probably going to be our answer, but we'll just check the last one. 2 diploid cells been produced. Don't need the spindle fibers for that. Do need the spindle fibers for that. So the answer is going to be C, the chromosomes shortening and thickening for this one. Okay. So which stage of mitosis would still occur even when it's present? The chromosomes shortening and thickening. Everything else that happens after that will not be able to happen because the spindle fibers are needed for the very next stage, which is to move those chromosomes from the equator to the opposite poles of the cell. Okay. And number 10, which statement describes a function of the cerebrum? So the cerebrum is usually associated with things like memory, intelligence, personality, and many other things. So let's have a look at the options. Remembering the rules of rugby. So that's memory, isn't it? So that's likely to be our answer, but we'll just check the other ones. B, increase in heart rate. That's going to be our medulla. C, coordinating movement of muscles when playing football. That's going to be our cerebellum. And maintaining balance during yoga, also going to be our cerebellum. Okay, so if you didn't get that one straight away, you need to look over your 3 parts of the brain and the function of each. Okay. Number 11, which row in the table identifies a hormone, its site of production, and its target tissue? Okay, so the hormones we've got are insulin. Then we've got glycogen. Now, I know it's tricky. We often get glycogen and glucagon mixed up, but glycogen is not a hormone. It's the storage carbohydrate. So we can eliminate B straight away from our consideration here. C is glucose, which again is not a hormone. It's our substrate, which begins the breakdown in respiration. So that one can also be eliminated. We can get rid of B and C. And D is glucagon, which is a hormone. So immediately we know that the answer has to be A or D. Now, where are those hormones produced? Well, they're both produced in the pancreas, okay? And if we look at the next column, the only one that can be correct based on the first 2 columns there is D. The glucagon is produced in the pancreas and its target tissue is, of course, the liver, okay? So D is going to be the correct answer there for number 11. Okay, number 12, a study was carried out to investigate the relationship between age and reaction time. The results are shown in the graph. Okay, so we've got males and females. We've got the reaction time in 1/100th of a second, and we've got the various age groups there from age 35 and under all the way up to age 66 and over. So which of the following conclusions is correct for these results? Now, when you get a conclusions question, you should go through every one of them and try and eliminate all the ones that aren't right and see what you're left with. Hopefully, it'll just be one by the end. So A, reaction time always increases with age for both males and females. So if we look at the data, reaction time does increase with age for females. However, for males, it actually goes down between the age 35 and under to the age 36 to 55. So A is not correct. B, reaction time always decreases with age for both males and females. Again, we know that's not true from the data. C, females have a lower reaction time than males in each year in each age group. That's not true. Again, we can look at the data quickly and see reaction time is greater. And males have a lower reaction time than females in each age group. And again, we can look at the data and compare each individual age group, and we can see that the males do, in fact, have a lower reaction time than females in each of the groups. So we're going to go for D there for question 12, just by process of elimination. For 13, the diagram shows the main parts of a flower. Which row in the table identifies structures G and the chromosome complement of the cells it, and this is the key word, produces? So let's have a look at our options here. For structure G, we can either go with the ovary or the ovule. Now, it's pointing to the ovary, okay, rather than the individual ovules within that area. So that's the structure. Now, what does the ovary produce? Well, it produces the haploid gametes. It produces ovules, which are haploid cells, okay? And the key thing is it's the chromosome complement of the cells produced. So that's going to be, 13 is going to be A, okay? For 14, the height of some cattle was measured and the presence or absence of horns was noted. Which row in the table is correct? Okay, so they've measured the height and the presence or absence of horns. So let's look at the feature, and then let's look at the type of variation, and then let's look at the type of inheritance and find the one that's going to be correct for this question. So A, horns is the feature. It says the type of variation is continuous. Now, it says in the stem of the question, the presence or absence of horns. To me, that says it's discrete. There's only two choices there, and not continuous. Okay, so A is going to be wrong from that point of view. For B, the feature is height. It says the variation is continuous, which for height we know is true because it has a wide range of values from a minimum to a maximum. And it says that the type of inheritance is polygenic. And again, we know that when we show continuous variation, that tends to be polygenic inheritance. So we think it's B, but let's just eliminate the others. C, height, discrete. Well, we know that height is not discrete. And D, horns, discrete. Now, we know horns are discrete. However, the type of inheritance for a discrete variation is going to be single gene, not polygenic. So the answer is, in fact, going to be B for question 14. In which part of a leaf are stomata found? Okay, so this goes to topic 2.5, that's transporting plants, and the leaf structure. Okay, the leaf structure section of that. So which part of the leaf are stomata found? The stomata are found at the very bottom of the leaf, which probably gives you a bit of a hint here as to what the answer is going to be, which means it's going to be the lower epidermis, okay? Stomata's right on the underside, so it'll be the lower epidermis, so 15 is going to be A. The palisade mesophyll is actually quite near the top of the leaf, the second layer down, where we've got those long cells with lots of chloroplasts to absorb lots of sunlight. And beneath that, we have the spongy mesophyll, which is all the air spaces, allows those diffusions of gases, and it allows water to evaporate out as well. And the leaf vein, which contains the xylem and phloem. But that is not the right answer. The answer is A, the lower epidermis. Okay, 16. The following factors can affect the rate of transpiration. So we know all our factors that affect the rate of transpiration. We've got increase in humidity here, decrease in humidity, increase in surface area, decrease in wind speed. Okay, they don't mention temperature here, but that's fine. It says which of these factors would cause a decrease in the rate of transpiration in a leafy shoot. So if we're looking at the factors that would cause a decrease in the rate of transpiration, we're looking for things like a decrease in temperature, which is not an option, a decrease in surface area, also not an option, a decrease in wind speed, which is an option, number 4. Remember, humidity is the one where it's the opposite. Humidity increase would cause a decrease in transpiration. So that's going to be 1 and 4 are our correct answers there. Okay, so if you got that one wrong or you're still not sure about that one, do look over the factors that affect the rate of transpiration again, and remember which ones, when they increase, cause an increase in transpiration, and which ones, when they decrease, will affect transpiration similarly. Okay, so the answer for this one is D, 1 and 4. Okay, remember humidity is the odd one out in terms of the factors. When humidity goes up, transpiration goes down and vice versa. Okay, number 17. Hemoglobin's ability to bind to oxygen is affected by the pH of the blood. The table shows the percentage of hemoglobin bound to oxygen at different pH levels. Okay, so we get 7.1, 7.2, 7.4, 7.5, and we've got the percentage. That's fine. The percentage of hemoglobin bound to oxygen when the pH of the blood is 7.1 is likely to be 2. When I was reading out those pHs there, what I did notice was it wasn't exactly linear, the increase. It was 7.1, 7.2, and then we missed 7.3 to go to 7.5, 7.4, sorry, then 7.5. So what I would do if I was doing the exam is I would actually put 7.3 in between them, and then I would get the number halfway between 60 and 69 there. So that'd be 64.5. Okay, 64.5 would be in here. And that tells us that we are going down by 4.5 for every 0.1 decrease in pH, which means that we then just need to take away 4.5 from 60, and that will give us 55.5. Okay, so our answer is going to be 55.5. Now, the mistake that students often make with this question is they go for 51 because they look at the decrease there, which is 9, and then take 9 away from 60 to get 51. But you've got to notice that the 7.3 has been missed from the table, and you basically have to fill that in just to remind yourself that it is actually going down in 4.5s. Okay, so that's going to be C for 17 there. Number 18, which row in the table identifies the substances absorbed into a structure found in the villus? So for number 18, what we have to do is we need to remember our lacteal and our capillaries and the substances that they absorb. So the capillaries absorb amino acids and glucose. Now, if we look at the two options for capillaries, neither of them says amino acids and glucose. One said fatty acids and glucose, and the second one says amino acids and glycerol. So we know that A and B are going to be incorrect. We then look at the lacteal, which we know absorbs fatty acids and glycerol. We look at C, amino acids and glucose, that's wrong, and D, fatty acids and glycerol. Okay, so the answer is going to be D for that one. So again, if you were not sure about this one, have a look over that 2.7 topic, the absorption topic, and look over the part about the small intestine and the villus and the lacteal and capillaries within, and remember the substances that each of them absorb. In number 19, the diagram represents part of a woodland food web. Which statement is true for the organisms in the food web? Okay, so we have to go through all the statements and decide which one is true. The owl and stoat are in intraspecific competition. So before I even look at the food web, I'm thinking intraspecific competition. Now that's got an A in it, okay? And so does the word same. Okay, that's my little hint for my class to remember that. So intraspecific is the same species. Is an owl and a stoat the same species? Nope, so it's not going to be A for that one. Okay, bank, vole and shrew are in interspecific competition. Okay, well, they are different species, at least this time, and interspecific is a different species. So now we have to look at the food web, and we look at the bank vole, and we look at the shrew over here. Now, are they in competition? They don't have the same food source there. Now, we don't know anything else about them from the food web. We don't know anything about all the other resources they need, but we know that they're not in competition for the same food, which means that this one is not going to be true. C, the ladybird is both predator and prey. Okay, so let's find the ladybird. And we can see the ladybird is in fact the predator of the greenfly, and it is the prey of the shrew. So it is both a predator and prey. Okay, so we think it's C, but let's just eliminate D as an option. So the greenfly is a producer. Well, if we have a look at the greenfly, they've tried to put this in, I'm assuming, because the word green, and we think of producers as green plants, photosynthetic plants, but it's not the bottom of the this particular food chain here. We can see we've got beech leaves from beech tree, and therefore is not going to be the producer. So D is incorrect as well. So C is our correct answer. All right, and it's good just to go through all the options and eliminate them, just for your own peace of mind to know that you certainly do have the right answer for that one. Okay, number 20. The graph shows the changes in population size of a predator and its prey over 20 years. Difference in the population sizes when each were at their maximum. Okay, so first of all, the predator is the solid line. So the solid line's maximum here looks as though it's around about 56,000. Okay, 56,000. Now, although that looks just like 56 on the scale there, remember the units, the units here on the scale, it says thousands. So that'll be 56,000. Okay, prey this time with the dashed line. We're going to find that up here at 72,000. Okay, so we've got 56,000 and 72,000. So we just do a quick calculation of 72,000 minus 56,000, and we should get 16,000. Okay, when we take away the 72,000 from the 56,000. And there we go. The answer is going to be B for that one. Okay, and again, students can get confused with this because they forget the thousands and they say 72, take away 56 is 16. 16 is not an option, but because it's in the thousands, hopefully people would realise and they would go for B for that one. Okay, next one, which of the following is a biotic factor which could affect biodiversity? So biotic factors, remember, are living factors, things which are alive, which could affect biodiversity. So soil moisture is not alive. Temperature is not alive, thankfully. Grazing. Now, grazing is eating grass. So grass is, in fact, a living thing. So that's likely to be a biotic factor. But we'll check D just to be sure, which is carbon dioxide concentration. And again, carbon dioxide is not alive. So the answer is going to be C, grazing for that one. Number 22. In an investigation into photosynthesis, a green plant was set up as shown and left in the light for 2 days. Okay, so if we have a look at it, leaf Q has been covered in black paper. So we can imagine what's going to happen to that leaf. Leaf R has absolutely nothing happening to it at all. So that one's going to photosynthesize brilliantly, I'm sure. Leaf P has got a plastic bag containing chemical to absorb carbon dioxide. So we are going to deprive leaf P of carbon dioxide. And again, you can imagine the effect that that's going to have. So leaves P, Q, and R were then tested for the presence of starch. In which of the leaves was starch present? Okay, now, you know, based on the way I've described the question, you're probably coming to an answer already, but P cannot photosynthesize and produce starch, which comes from the sugar at the end of the carbon fixation stage without carbon dioxide. For leaf Q, the light reaction stage cannot take place because we're not absorbing that light energy via the chlorophyll in the chloroplast. Okay, so therefore we can't even get started on photosynthesis with leaf Q. Whereas leaf R has everything it needs and therefore is going to photosynthesize, produce sugar, and that sugar can be converted into starch. So the answer is going to be B for this one, leaf R only. Okay, number 23. The table shows the results of an investigation into the effect of temperature on egg-laying mites. As the temperature increases, the average number of eggs laid per female per day, and please note when they put something in bold in a question, you must take note of this, okay? This is per day. Now, what you'll notice as well is that in the table here, we've got the temperatures and we've got the average length of egg laying period in days. So that means you're going to have to do something with this information because, again, it's in bold in that question. We've also got the average number of eggs laid per female during the egg laying period in total. So what we have to do for this is we have to divide the total number of eggs laid by the number of days that they had as their egg laying period. So the first one would be 108 divided by 36. So that's going to be 3 for this first one. The second one is going to be 108 divided by 27 days. Again, this is so that we get it per day. That's going to be 4. And the last one is 108 divided by 18, which is going to give us 6 per day. Okay. So as the temperature increases, okay, the temperature is increasing along this way. As the temperature increases, the average number of eggs laid per female per day actually increases. It goes up. Okay. So it's going to be A is the answer for this one. And again, the common mistake for students is they don't realise they have to do it per day, or they're not sure how to work that out. And so they see 36, 27 and 18 and think that's actually going down. Okay, but if we do it per day, because this one here at the end has the fewest days, then they actually have the most eggs laid per day. Okay, number 24. Which row in the table describes the changes that take place when fertiliser leaches into a river? Okay, so the first thing that's going to happen when fertiliser leaches into a river is that the algal bloom is going to form, right? So the algal population is going to increase. That means our answers can only be A or B. What's going to happen to the amount of light that's able to get into the water then? Well, that's going to go down, and that doesn't help us a lot because both A and B are both decreased for that anyway. For the next one, the bacterial numbers. Now, we have all this algal bloom suddenly forming in the water, but eventually that is going to start to die, and our friendly bacterium are going to come along and start to decompose that dead, rotting material. So the bacterial numbers are going to increase. And then finally, because the bacterial numbers increase and they carry out respiration, the oxygen levels are going to decrease within the water. Okay, so the answer we're looking for 24 is A. Okay, next one, we've got paints containing toxic mercury compounds were used to paint ships. As a result, mercury entered the marine food chains. The concentration of mercury compounds in the tissues of the organisms in the following marine food chain was measured. So we've got plankton eaten by aquatic insects, eaten by small fish, eaten by large fish. So which bar on the graph represents the mercury concentration of the small fish? So we need to remember our bioaccumulation here. As we go up the food chain, the amount of the initial chemical absorbed increases in concentration with the organisms further up the food chain. So that should hopefully help us here when we're looking at this. Okay, so let's have a look. We've got organism A is the highest, organism B is the second highest, organism C is the third, and organism D has very little mercury. Now, because small fish is third in the food chain, that means it will have the second most. The large fish will have the most, the small fish will have the next most. And if we look at the bar graph here, B is the one with the second most, so therefore B is going to represent the mercury concentration of the small fish. Okay, folks, that's our paper done. I hope that was useful for you, the little walkthrough. Let me know if you have any questions or queries in the comments, and I'll see you next time.